go ahead and get started. Per the usual, enjoy the memes. I hope you do. I spend a lot of time finding these, just so you know. But I just do that because I want to, so it has nothing to do with you guys. All right, we're going to start talking about today's material, chapter 15. Pretty much just going to go over Friday's lecture and today's lecture. But if you guys have questions about other Exam 3 stuff, which isn't a whole lot yet. Speaking of, Exam 3 opens in 2 weeks, so just so you know, these tests are kind of non-stop. So my super session is actually like 3 sessions from now, if that helps you feel a little nervous about it. And that may seem advantageous. You may go, well, that's fine because we can't cover that much material in the next 2 weeks before the test. You'd be wrong. We will. We will cover a lot of material in the next 2 weeks. Per my opinion, test 3 was the most difficult for me personally. For other people, it may be different. I always hesitate to say that because then people like start freaking out and then they're like, test 3 wasn't even hard, are you an idiot? And I'm like, well, yes, but for me that was the most difficult material. So I just put that out there so that if you want to take my opinion, test 3 was the most difficult. I only put it out there so that you are aware that while we have less time, ergo less material, it's still a lot of stuff. And you need to know it very well. Again, I cautioned in my last session, don't get cocky, don't go, oh, Punnett squares really aren't that hard. And I learned a little bit of genetics in high school, or if you took Bio 1010, you've already seen a lot of this stuff. It's going to get a lot harder real fast, like especially the next couple of days here. I guess we don't have Friday, but like Wednesday and then next Monday, it starts to just go through the roof, okay? So stay on top of stuff. Got to pass the roll around. You know the drill. 8:30 on the front, 9:30 on the back, just your name. Do you have any questions before we get started? Gonna assume it's a no. All right, so our biology fact for the day. Here at USU, we conduct transgenic research that lets us harvest spider silk from goat milk. So since we're talking about genetics, what they do is they take the genes that code for silk in spiders and they plant those. They basically cut out the genes for milk in a goat and they put in spider silk genes, and instead of milk, the goats put out spider silk. And then they actually can use those to form artificial tendons for restorative surgeries is probably a better term. So I just think that's super cool, especially because it's something we do here at the university. We can milk goats and get spider silk and make Achilles tendons, which I think is super cool. In case you thought genetics was boring. Oh, yeah.
Are you going to cover segregation?
Briefly. If I don't cover whatever question you may have, feel free to ask, though. Other questions? No? All right. Don't misinterpret this. I don't mean just study for 15 minutes. I mean make them good 15 minutes, right? If the first 15 minutes of your study session is terrible, if you're checking Facebook and texting people and switching stations on Pandora and getting up to get a snack and going to the bathroom all within 15 minutes, A, you probably have done zero studying. I'd be surprised if you could even do all of that in 15 minutes. But B, you've already set yourself up for failure, okay? So the first 15 minutes of your study session should be real good, okay? Now, I understand if you've slotted 3 hours for studying that day, hopefully it's not 3 consecutive hours. That's a bad idea. I did like 1.5 consecutive hours earlier today, and I was like, I can't focus anymore. So I broke it up, right? I'm going to do some more tonight. I'm not just going to, call it good, but break it up, make the first 15 minutes real good. If you got to take a break after 15 minutes, that's fine. but at least make the first 15 minutes really solid, okay? It'll make a difference. I know you guys think I just do these study tips out of like rote habit, but they make a difference. So if you try even some of these, you'll find one that works and it'll help you out. All right, so let's start off talking about segregation. Well, so what am I looking at here in this diagram? Meiosis, okay, that's one answer. What else? There's a few things going on here. I heard independent assortment. Oh, it's not letting me write. Where's my thing? I might not be able to write today. What is going on? Uh-oh. Is anybody better at PowerPoint than me? It's never done this. I may not be able to write, so sorry for people watching the Panopto. The first step there, possibility 1 or 2, that's independent assortment, right? Somebody define independent assortment for me. Not very difficult.
The random likelihood that homologous pairs will line up in whatever order along the metaphase plate.
Perfect. Random chance, 50-50 chance that the homologs line up in one order or the other. Basically just meaning, both blue on the left or one blue, one red on the left, one red, one blue on the right, right? 50-50 chance. What does this do? Why do we even care about independent assortment? Causes genetic variation, right? So it's one of the things that allows an increased amount of genotypes and therefore phenotypes and therefore genetic diversity, right? Which is always good. All right. I guess it says right there, the middle part there is metaphase 2. But what has happened to all those bottom rows there? What have they undergone besides meiosis? Segregation, right? So what is segregation in genetics terms? Okay, two genotype, kind of. Two what? Two alleles, right? So I really can't draw, and it's driving me nuts. This half segregates from that half. This half comes over here, and that half goes over there. Again, apologies to the Panopto watchers. The left half of the blue one goes to the left cell, and the right half of the blue one goes to the right cell, right? They've segregated from each other. That's what segregation is.
Is one chromatid the same thing as an allele? In this case, each chromatid, where we're talking about like sister chromatids in one chromosome, contain one copy of an allele. So again, I can't draw, but let's say the gene for hair color is right here on this blue chromosome, sort of towards the tip, right? The other allele right next to it, the other chromatid making up the same sister chromosome next to it is also going to have the hair color gene right here. So when we say the alleles segregate, this chromatid goes to one cell and this chromatid goes to one cell, but they contain identical copies of the allele, so the alleles also segregate because they're stuck to the chromosomes. Does that help? I saw another hand.
Yeah, so you mentioned today that segregation isn't always independent assortment, or maybe the other way around. I just didn't really understand that.
He mentioned independent assortment isn't segregation.
Isn't always segregation, or you remember that?
There's a clicker.
It was like a clicker question. It's like true or false. Segregation is always going to be independent assortment, and vice versa. And one of them was right, one of them was wrong.
Does anybody remember the clicker question? I feel like I can't.
So independent assortment is always segregation, but segregation is not always necessarily independent assortment.
Okay.
I just don't understand it.
Well, if somebody want to help me out, I wasn't actually there for this clicker question. I still don't know what we're talking about exactly. Say it loud and proud.
So independent assortment is basically just how the homologous pairs line up, whether it's the reds are on one side or they're on the different sides and they separate. Whereas segregation is the alleles separating from each other into two different daughter cells. Is that right?
Yeah.
Does that help?
Yeah.
Good. See, you guys don't even need me. What am I even here for? I'm here for the memes, right? All right. Other questions?
Segregation is happening in crossover as well, right? Once they separate.
You mean after they cross over, do they segregate?
Yeah.
Yes. So the crossing over, when does crossing over happen? I heard some mumbling. Metaphase 1, what part of metaphase 1? Or not metaphase 1, sorry, meiosis 1. What part of meiosis 1? Prophase 1, obviously, right? So they do cross over in prophase 1 of meiosis 1. Then, after they've done the whole thing and finished meiosis 2, then they segregate. Yeah. So they've already crossed over quite a few steps earlier. But that again just increases the genetic diversity because now we're segregating all over the place after having crossed over and sorted independently and just, so that's why everybody looks different, right? I feel like that's my catchphrase with genetics. Everybody looks different because genetic diversity, right? Because who knows, right? There's no way, well, there's a lot of ways. That sounds terrible. It is incredibly difficult to predict exactly what's going to happen. That's why you can't go to like a genetic counselor and say, all right, so I want to have a green-eyed child with light brown hair who's 6 foot tall and really good at soccer and weighs about 200 pounds. You can't pick and choose these. Nobody can because it is random, right? There's some controls. We don't do this in humans. That seems a little bit unethical. But we can pick and choose and sort in a lot of ways. First of all, breeding, right? That's how we got pugs. Pugs are not environmentally fit for survival. They have to have surgery just to breathe, right? So nobody exactly went, let's make it so that they can't breathe, but that was, a side effect. But we do all this, picking and choosing and breeding. And we can actually just go in and specifically, now that we have the technology, just straight up cut out genes like with the goats and just put genes in and do whatever we want, right? So outside of humans, yeah, we actually probably could go, I want a dog that is exactly this size and exactly this and this and this and this and this. And you would cut up all the genes and mix them around and whatever. Other than that, it's totally random, right? Unless you're trying to play God, it's just going to happen how it happens, okay? So that's the idea behind independent assortment, crossing over, and random fertilization, right? All of those are for genetic diversity. And then segregation is just, we have to divide up the alleles, right? That's kind of the whole reason that we did any of this is to get those very bottom cells. All right. I don't want to spend a lot more time on this because I have a lot of practice problems for like Punnett squares and pedigrees, and that's what I want to devote most of the time to. But does anybody have a quick question before we move on? I don't mean to like make you feel like you can't.
Are the questions going to incorporate like the ratios?
Like test questions?
Yeah.
It's definitely possible, yeah, that he could ask you what would be the genotypic ratio in this cross, or what would be the phenotypic ratio. So you have to, A, know how to find either ratio, and B, know how to differentiate which one's which. Yeah, so we'll look at that as well. Other questions? The question was, will we have to know how to do ratios? One of the things that I got on those midterm evaluations was that I don't repeat the questions into the microphone, which is true. So I'm trying to do better at that. I read them. You guys think I don't? Some of you were mean. Only a few of you. Incomplete dominance, right? What is incomplete dominance besides what's on the slide there? Somebody explain it to me, as if I knew nothing about genetics. Some of you think I don't.
Basically, like the two genotypes, neither one of them are dominant to take over the other, so they kind of mix.
Okay.
Make pink instead of red or white.
Yeah, so they're incompletely dominant. Neither one beats the other. Neither one, they're both technically dominant traits. So neither one, there's no recessive trait in this case, but neither one is so dominant that it like pushes out the other. So they mix together, okay? Incomplete dominance is always a mixture of the two, right? So red flower, white flower, you get pink flower, right?
You guys see how that happened?
So then why in Mendel's experiment with the white and the purple flowers, why did this blending happen? Why didn't it just cancel out the other? You know? Not cancel out, but kind of
Right.
cover over.
They weren't incompletely dominant traits. One was dominant, one was recessive. And so it just depends on what trait you're looking at, and it'll differ by species, differ by trait, differ by all kinds of stuff. In this specific flower, petal color is an incompletely dominant trait. Whereas in pea plants, flower color is a dominant or recessive trait. Does that make sense? Yeah.
What's the difference between that and codominance?
Aha, we'll get to codominance in a second here. Very good question. I compare this to toddlers, right? And each one wants what each one wants, right? One wants to go to McDonald's, and one wants to go to Wendy's, and neither one of them will be okay with the other. If you go to McDonald's, one of them will cry, right? It's just going to happen, okay? So where do you go? Burger King, right? You meet in the middle, and you don't go to either, because one way or the other, they're going to cry, right? So you just say, you know what, we're not going to Wendy's or McDonald's, right? We're going to Burger King, and everyone's going to be sad, because I hate Burger King. Any Burger King fans out there will fight after. It still won't let me draw. I think the pen's not even synced to it. Why am I even still carrying it? Okay, questions on incomplete dominance?
You can't consider this blending, or can you? 'Cause is there a difference between, because they say that blending doesn't exist, or Mendel's theory of blending wasn't correct?
I don't know. That kind of all breaks down to semantics, blending, mixing, crossing. You got to be careful what you're choosing, but basically it's a blend of the two traits. Whether it's whatever word you want to use for it, it's just the traits don't. I can't think of a different word besides blend or mix, but they mix together and they are an intermediate, I guess.
You could say like the gold show like halfway.
Yeah. So the red kind of shows through, but the white also kind of shows through. And if you were to mix red and white paint, you get pink, right? So something like that.
So if you're doing a calculation for this or the table, and you got a red, or you found that you had gotten a red, do you still declare it's an incomplete dominance because it still has the option of being able to get pink, even though you're trying to figure out red?
Yeah, I mean, you'd have to have gotten enough offspring to see, oh, I got red and white and pink. If you got all red, you'd probably just go, oh, red's dominant. But if you crossed a red and a white and you got some red, some white, and some pink, then you would go, okay, I'm definitely seeing incomplete dominance here because two of them mixed together. Does that help? Okay, I think I saw another hand, unless I already answered it. All right. So this is what it would look like in a Punnett square, right? This is just another way of denoting alleles, right? So we've been using A's and B's and C's and whatnot. Some people prefer to use the letter for the trait that you're looking at. So in this case, C for color, right? Because then you can say the color is red and the color is white. So the little superscript R and W are for the actual colors. The big C is this is color, and then the superscript is which color it is. Anyway, so you take a red and a white and you cross them together, and you're going to get half red, half white. But mixed together, because this is incompletely dominant, is pink, right? Then you cross a pink with a pink, and that's your Punnett square for what you can get. So you can get a red, you get two pinks, or a white, right? Does everybody see how that Punnett square works? It's not technically any different from any of our previous Punnett squares, just that since they're both dominant, you can't say, you know, it's a 3-to-1 ratio, it's a 1-to-2-to-1 ratio, right? Does everybody see what I'm talking about? Mostly I just wanted you to see that they're not vastly different. The only difference is that mixing.
So I've noticed a lot these questions come out quite frequently for this chapter on like the homework assignments and everything. Is it just the quickest to write out the Punnett square to solve the problem?
I would say for something like this, yeah.
If you're dealing with like 5 or 6 alleles, where you're going to have like 256 possibilities, some mathematical equations probably help, but on one of these, yeah, it's probably quickest just to make your Punnett square. I can't remember whether or not Dr. Pogorski will show you the mathematical way to do it. It's faster and easier, but I don't want to tell you about it because it's a secret. No, if we don't talk about it in this class, then I don't want you to worry about it, because there is a mathematical way to do this, because usually genes are thousands of genes, right? So there's a mathematical way to do it, but until we talk about it, I won't give anything away, right? But there is. That sounds terrible. There's a better way to do this, but I'm not going to talk about it. I just don't want to waste time because I can't remember whether or not Dr. Pogorski explains it. So anyway. All right. So this is just a quick slide about the idea of multiple alleles, okay? So for example, in blood types, the allele is I, right? That's how we denote it. But then there's an A-type allele, a B-type allele, and then the recessive is technically an O-type, but there's no need to put I-O, partly because it would look like I-not and people would get confused. But it's the recessive trait, so you just put a lowercase letter, right? So the trait that it actually produces is an A-type carbohydrate, a B-type carbohydrate, or no carbohydrates on your blood cells, okay? So this is just showing that not everything is A's and B's, right? Not everything is just one or the other allele, right? There can be multiple alleles, and we'll see that even going forward. But blood cells also express codominance, right? So somebody asked, what's the difference between incomplete dominance and codominance? Who can answer that for me?
Codominance, you still get some of both, and you get separate traits that are manifested. In incomplete dominance, you just have kind of a mixture.
Perfect. So codominance, rather than mixing together and getting some kind of intermediate between the two, you get both showing up, right? So in A-type blood cells, you get all A-type carbohydrates. In B-type blood cells, you get all B carbohydrates. In AB, or the codominant combination of A and B type blood cells, you get A and B carbohydrates, right? You don't get like a triangle circle thing. I don't know, what would that be? An oval? They don't mix together, they just both show up, right? I'll have some more examples of this later on. And then in the O blood type, you don't have any carbohydrates.
So with codominance and like flowers, instead of being pink, it would be like spotted red and white.
Yes, you would get red and white spots showing up instead of mixing them together. It's a perfect example. Just a sidebar, because I think this is interesting. You have your 4 blood types, but then there's also positive and negative, right? You guys have heard of like I'm O negative or I'm B positive or whatever. That's just whether or not a special protein is on the blood, right? So this is just talking about the carbohydrates. A, B, AB, or O is the type of carbohydrate there, or in the case of O, no carbohydrates. But then if you have special proteins, they're called like rhesus factors, not like Reese's, but rhesus factors. Those determine positive and negative blood types, which is something else. Other questions? So the circled one, right, is the codominant. Well, the trait is codominant, but that's the one expressing codominance, right? So, I found this on the internet. This is not how pandas work, right? But I thought it was kind of a fun way to look at it. Okay, so don't go thinking I'm an idiot. But we take a black bear and a polar bear. They're both, black is a dominant trait. White is a dominant trait, right? So it's big B, big W, big W. If you cross them, you end up with black and white, right? So is this incomplete dominance or codominance? Codominance, right? There was no mixing. Both traits appeared. Now, if this were incomplete dominance, what would their offspring look like? Probably like grayish, right? There wouldn't be any spotting. It would just be kind of an overall gray between black and white.
So then would it be a 1-to-1 ratio? Not like a
For the offspring, right here on this?
If it was incomplete, you would have a
I think-
a
I think it would
1-to-1
just-
ratio.
Oh. If it were incomplete dominance, would you have a 1 to 2 to 1? Yes. So you would end up with probably like a
Big W, small w.
like a black one, 2 gray ones, and a white one.
and a white one.
Yeah.
Can you have more than 2 traits in codominance, or is it just only 2?
Not that I've ever heard of. Doesn't mean that there isn't, but as far as we go into this class, we just look at basically 2 traits. Probably could be more. I don't see any reason not to.
Is there any special ratio for the codominance?
At least right here, no, it's a 1 to, just a 1. They're all just black and white.
So what about the blood types?
The blood types, what would the ratio be? I guess I'd have to draw out a full Punnett square. I don't think there's any like special ratio. You would do the same equation that I won't tell you about, or draw a Punnett square, same as you would draw a Punnett square, and you would just have to manually go through. And I don't think there's any special difference between the ratios per se.
If you were to draw a Punnett square of this, would you have to put, because you need to have 2 different gene sequences, genotypes, and they can be for like A and B, so would you have to put them separately, or would they just be the same?
I wish I could show you. I can't draw on this thing. Basically, quick answer, no. So you would just have like IA, IB, or little I, and then depending on the 2 parents that you cross, if it's like an IA, IB, so AB blood type, you would just have IA, IB. I guess I could go old school, right? So if you're crossing like oh, that's terrible. AB blood type with an A blood type, you would basically just have, I guess,
I've got a rare marker if you want to use this one.
yeah. Are you guys impressed? Let's hope it's better. Oh, it is. So you'd have like IA, IB. So see how I'm just making the Punnett square the same way I always do? So nothing's really changed. Just if I were to have like, you know, little I, then it's just going to be, I guess, right? So you make them the same way. You just have to keep track of more traits, I guess, and you have more combinations. All right, other questions? All right, so we decided that was codominance. What about this? We take I want to say red flower and a white flower, and I'm assuming that's pink. It's like a weird pink. What is this? Incomplete dominance. This one's kind of lame because it's basically the same as the first example we looked at, but it's incomplete dominance, right? You don't get red or white. You get somewhere in between pink, right?
So in this one, the white is just not dominant at all.
They're just, the term is incompletely dominant. So they both are expressed, but because they're both expressed in equal amounts, neither one kind of trumps the other. They blend together. So they are both dominant traits. They are incompletely dominant.
How do you determine which ratio goes with which dominance? So like 1 to 1 or 3 to 1, or I don't know if that makes sense. Is it set or does it vary?
It varies by whether it's complete dominant, or sorry, codominance, incomplete dominance, or dominant recessive.
So dominant recessive, you usually see, well, I guess that depends on how many traits you're looking at too. If you're looking at 2 traits, then it's like a 9 to 3 to 3 to 1. If you're looking at 2 traits, or I guess 1 trait, you're looking at like a 3 to 1. So they each vary, and they each have, there's not a short answer to that, I guess. Sorry. I saw another hand.
Oh yeah, we're just looking at the F1 generation right here, right? And then the next F2 generation would be the red with the 2 pink and then the white.
Yeah, so then if you were to, I should have kept your marker. If you were to take, so now a pink with a pink, right? You're going to have a red, 2 pinks, and a white. Yeah. So that's, I think we saw that in this one. So what's the genotypic ratio on this? 1 to 2 to 1, right? Okay, so it varies by what kind of dominance you're looking at, how many traits you're looking at, all that stuff. Does that answer your question, whoever asked it? Okay. Other questions?
Yeah, like say that red is dominant and white is recessive, what would that be?
So if red were dominant to white, then they would all be red in this case, because anytime you have a big R, then you would see red.
What about
Were this to be codominant, we would have to use a different letter here so that we don't confuse ourselves, because they're both dominant, so we would use like W, right? And then you would end up with RW, 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 RW. And so they would all be red and white.
So you can't have just one codominant, you have to have one.
Right, because co meaning both, so they each are dominant to each other, I guess, so they both show up. So there does have to be, there's no such thing as like a single codominant trait with like a single recessive. They're both dominant, they both show up. Other questions?
Can you go over how you know what the ratio is?
So, I'm going to use my black marker because it's a lot better. So let's cross like a big R with a big R, little r. So we get R, R, and R, little r. So we have for like a genotypic ratio, you literally just kind of count up. So you say I have 2 of these and 2 of those. So I have a 2 to 2, or I guess 1 to 1 if you want to simplify, ratio, right? Or you could do heterozygous. So you have a big R, big R, big R, little r, big R, little r, little r, little r. So you have 1 of those, 2 of those, and 1 of those. So that's like a 1 to 2 to 1. Okay? But again, these have all been genotypic ratios. What if I ask you about the phenotypic ratio? Let's say this is red and this is white. And red is dominant to white. Okay, we're not talking codominant, nothing like that. What's the phenotypic ratio here? I heard a lot of people say 3 to 1. How'd you come up with that? Because that's red, right, with the dominant. That's still red because the big R is dominant. That's still red, big R is dominant. And that's white. So that's the genotypic ratio. The phenotypic ratio would be 3 to 1. So you just have to count out. And you have to know whether you're looking at genotypic ratio or phenotypic ratio, because they're going to be completely, most of the time, completely different from each other, right?
If they were incomplete dominance, though, the phenotypical and genotype ratio is the same.
Let's think this out. If this one were incompletely dominant, is that what you're saying?
Yeah, so it would be red
So this, because we're saying R is white, so this would be red, these would be pink, and that would be white. Yeah, so it's a 1 to 2 to 1 in that case.
So
Both of them would be in that case. So in that specific case, the genotype and phenotype happen to be the same ratio. Most of the time that won't happen.
I was curious, why would this not be complete dominance?
Complete, you mean codominance? Or just dominance?
Dominance, yeah.
Because in dominance, R would be dominant to little r, right? Or red would be dominant to white. So we would only get red.
Okay.
But because you got that mix, incomplete, yeah. Are you colorblind?
No, I
Oh.
was not.
Because I am. So I look at this and I'm like, I assume it's pink? I don't know. That's why it's important to read. I have to read the question before I just start like looking at pictures. Otherwise, I don't know what's going on.
So if they were to ask like the phenotypic ratio for the one on the left, would they ask that because it's all the same? Like how would you?
The phenotypic ratio here, if we're saying that this is red and this is white, but red is completely dominant, then the phenotypic ratio would just be 1, because they're all red, right? What would the genotypic ratio on that one be, though? Huh? This one, genotypic ratio? 2 to 2 or 1 to 1 if you want to simplify, right? So we get 2 big R, 2 big R, little r. Okay?
Should we worry about simplifying the genotypical ones in the exam? Like is
Well, because it's multiple choice, you won't see both of these because they'd technically both be the correct answer, and so he won't do that. So if you see 2 to 2 or 1 to 1, know that they're the same thing. Other questions? I think it's more technically correct to put this because that also helps us keep track of how many offspring we got. I just, because I've had math drilled into me since first grade, I just simplify automatically, but I think this is more technically correct. All right, we're running out of time. I still have so many things to cover. These are Andalusian hens, okay? Well, Andalusian chickens, because they can't both be hens. It doesn't work in biology. We have a black one and a white one. You cross them, we get a blue one. You cross them again, you get a black one, 2 blue ones, and a white one. What are we looking at? Incomplete dominance, right? Anytime, I about drew on this thing with the freaking Expo marker. Put that away. Anytime you see a mix, so anytime you see blue, when it should, I don't know why it's not gray, but blue, we've mixed them, right? As soon as you see mix, throw every other option out. If the two phenotypes have blended together and you get an intermediate that's neither of the first two, incomplete dominance, just click on it and move on, right? Don't even, nothing, right? If they've mixed, incomplete dominance, and just let it go, right? It is that easy. All right, and we have a who knows what color cow. Technically it's roan. I only know that because they do a lot of experiments and things like this, where you do like a roan cow with a cream cow. I don't know. Aren't they all cream cows? You get roan spots and technically white spots, but it's kind of like the base color. So what is this? Codominance, right? We got both of them showing up. They're not blending together. They're both displaying. Okay? Questions up till here? All right. So let's talk about pleiotropy. What is pleiotropy? One gene, many traits. Okay? Pound that phrase into your head. Pleiotropy, one gene, many traits. Okay? The example from class, sickle cell anemia. There is one gene that determines whether you have normal, spherical, well, sort of disc-shaped blood cells or these kind of sickle-shaped blood cells, right? One gene. If you have that recessive trait, you have sickle cell anemia. But if you have sickle cell anemia, your red blood cells break down, they clump up, you get accumulation in your spleen, spleen damage, damaged organs, brain, pain, heart failure, anemia. You get just a whole cascade of traits, right? So that single gene led to all of these traits. Pleiotropy. Before you ask questions, I'm going to compare it against polygenic inheritance is then what? Many genes, one trait, right? It's just the opposite. Polygenic inheritance, many genes, one trait. Pleiotropy, one gene, many traits, okay? So I find it easier to define polygenic inheritance first because polygenic, many genes. And then I go, well, many genes, one trait, so then pleiotropy's got to be one gene, many traits, right? So skin color. This is a hypothetical example. There's actually way more genes involved. But there are 8 possibilities on the top and 8 possibilities on the side there because of the heterozygotes that we've crossed, right? And you can get all kinds of intermediates, everything from, what's the word where you don't have? Albino. Why couldn't I think of it? Albinism, or just very, very, very dark skin, right? There's not a term for that. Black. You can be on either end of the spectrum. More than likely, you'll kind of be in these intermediate ranges, right? So I'm like right in between these two. I'm very white, but I'm not albino. So see how we have an A gene, a B gene, a C gene, but each of these contribute to your skin color. Many genes affecting one single trait. Okay?
Why does it say 1/8? Like, I get that there's 3 genes, but why are we looking at something that says 1/8, and why are there 8 options?
I think it's just saying of the 8 possibilities, this is one of 8, this is one of 8, this is one of 8. So it makes up 1/8 of the possibilities that you can get from like the sperm. And then you can get 8 possibilities from the eggs, but then you have 64 total possibilities. So it's just showing it's one of 8. Does that make sense?
Not really.
No. I can't draw on it, but there are 8 possible combinations you can get from the sperm. So that first top left, just the 3 empty circles, 3 white circles, however you want to look at them, that represents 1/8 of the possibilities.
Okay, so what's the relationship between the 1/8, that one possibility, and the 3 genes that are in the...
So the large A and the little a can give you 2 possibilities. And then big B and little b gives you 2, and C, and then you times them. 2 times 2 is 4, and 2 times 2, or 4 times 2 is 8.
Yeah. So you take the number of genes by the number of possibilities. Well, so you take the number of alleles, so 2, and then to the number of genes, so 2 to the 3rd, 8. Other questions? I thought I saw another hand. No? All right. So, do we have time? Let's look at pedigrees. The first thing you need to absolutely drill in your head, squares are male, circles are female. Filled in means affected. Not dominant, not recessive. Everybody right off the bat goes, okay, it's filled in, so that's the dominant trait. It just means it's the trait that we're looking at, right? Color blindness, height, hemophilia, Parkinson's, whatever you're looking at. The filled in, means that they are displaying the trait that we're looking at. Non-filled in, not displaying the trait, right? Mating pairs, male and female, is a horizontal line. Their offspring have a vertical line coming off of them. Hopefully there's not a horizontal line between those, at least if we're looking at humans. That would be bad for reasons you'll learn in a further advanced genetics course, but basically it's bad. Inbreeding is not good. You get a lot of problems. Okay, so this is what a pedigree for a dominant trait will look like. Okay? So, if I hadn't told you this was dominant, and if these W's weren't here, how would you know? It's kind of like a logic puzzle. How do you figure it out?
You look at the children and see what the outcomes were between them.
That's definitely a way to look at it, but run me through it.
Because there's 12 shapes and half of them are affected.
Okay, that's one way to look at it. You can count up how many shapes, count out how many of them are affected. If it's about half, then you're probably looking at a dominant trait. If it's significantly less, then it's probably recessive, right? I hesitate to use that method only because that's only 3 generations, when these girls probably have thousands of ancestors, right? So what if they're the only like 6 that have it and the other like 10 million don't, but we just didn't see that? So that works, but I would be careful with it. How else can you do it? Do you guys notice a pattern? Look for patterns. Have many of you done a logic puzzle before? They're all patterns, okay, and logical deduction. If you take like an IQ test, stuff like this would show up. All right, I'll ruin it for you. Do you notice that every affected person has an affected child? Always. It means it's a dominant trait, right? If I have it, at least one of my offspring will have it, right? Because it's a dominant trait. So let's look at the opposite then, a recessive trait. These are usually easier to spot, but do the same thing. Look for the pattern. How do I know it's recessive?
It's recessive. It's all the ones that are affected have the recessive allele.
Yeah, but if I hadn't given you the T's, how would you know? Okay, so as soon as you see 2 unaffected having an affected, shut it down, it's recessive. If you see 2 empty, like an empty square and an empty circle, having a filled in circle, recessive. Right off the bat, you don't have to look any further. It does not matter. That is recessive. Okay? That's a rule. All right, running out of time here, so I'm just going to keep going. Hemophilia. Okay, I took a very long time making this myself and Googling all of their names. Okay, not really. It was the 3rd thing that popped up under Google Images when I put pedigrees. Dominant or recessive trait? Recessive. You probably know that just because you know that hemophilia is not all that common, so it's probably a recessive trait. But how else do you know that? Okay, Albert and Victoria don't have hemophilia, but Leopold does. Has to be recessive, right? So if we're saying hemophilia is H's, then big H, little h, big H, little h, right? Because that's the only way you could get little h, little h. That's the only way you could get hemophilia. Question?
Isn't hemophilia a gene-linked trait?
It is a sex-linked trait, so we'll talk about those, I want to say, Wednesday. So that also factors in, right? Notice that they're all males, right? So it is a sex-linked trait. It's a sex-linked recessive trait. But that's good. I'm surprised you noticed that. So you would normally notice that it's a sex-linked trait if pretty much only males display it, right? Color blindness, same thing, right? 90% of colorblind people are male because it's a sex-linked trait. And it mostly affects males. But we'll look at the mechanics of that soon. All right, let's practice. This is Pedigree 1. It's autosomal, meaning it's not sex-linked, right? It doesn't matter gender here. I've covered up most of the genotypes, and I've covered up whether it's dominant or recessive. Go ahead and first tell me dominant or recessive.
I got some people saying dominant, some people saying recessive. Who says dominant? Some of you started to chicken out. Who says recessive? All right, how'd you know? In generation 2, 4 and 5 are unaffected, but then in generation 3, number 1 is affected. Boom. Recessive off the bat.
So in a test, we would just ask, is this recessive or dominant? Because isn't the white dominant and the black is recessive? Like, isn't there both?
Filled in just means affected. Not dominant, not recessive. It means displaying the trait we're looking at. So if this is color blindness, filled in just means I'm colorblind.
So we're looking at the filled in ones to figure out what they are?
Kind of. You look at the relationships. So see how 4 and 5 in that second generation there are empty, so they're not affected, but then they have an affected child. If they're unaffected and have an affected child, it has to be a recessive trait. That's the only way that could happen. I saw another hand.
So I just wanted to clarify about, are we, when we're looking at recessive or dominant traits, are we looking at primarily generation 1 or over 2, or are we looking at both?
Like if you want to decide, is this recessive or dominant?
Because I thought it was recessive.
It is
No,
recessive.
I thought it was dominant.
Oh. Yeah, you probably went, well, 1 and 2,
Yeah,
one of them has it, so it's dominant.
yeah.
There's not a generation that kind of like trumps the other. I would just look for either 2 unexpressing, 2 unaffected, having an affected, or an affected and an unaffected. If this were dominant, then at least some of them should have had it, right? So those are the 2 patterns to look for, either 2 unaffected having an affected, or an affected and an unaffected, or an affected and an affected having affected, right? That's how you differentiate.
So even without like the 3rd generation, do we still tell this?
I would say I would be fairly confident by looking at generation 1, mixing 1 and 2, and how none of them have the trait. I would go, none of them have it, pretty likely that it's recessive. Could have just been bad odds though, so keep going. But I guess if that had been all you were given, you could have said with a fair degree of confidence this is recessive, but as soon as you see this, 100% you know it's recessive. So keep going till you find one that you go, nope, it is for sure dominant or for sure recessive. Don't just look at the first ones and go, well, there's an affected one, it's dominant, right? Don't do that.
So if generation 2, number 4 was dominant, like if it was filled in, then that would make the trait dominant, right?
Yeah, it would have been a dominant trait because if 2-4 had been filled in, then there's no way that could have been a recessive trait. You would have to, I don't know how else to put it. You would have to have both empty and then unaffected for it to be recessive. Okay, so we're pretty much out of time, probably more than out of time, so I'll just reveal it now, but we were going to go through and look at each of these, right? So, but take a look at it before you run off, make sense of what you're seeing here, and then note those last 2 are blank because we don't have enough information to decide if this is homozygous or heterozygous, right? We'd have to look at their children to see what else comes out of it, okay? So if you just see like an A blank, it just means there's not enough information. Don't panic and go, what is it? We don't know it yet. All right, guys, thanks for being here.